Good afternoon, students. Today we are going to discuss about the chronic inflammation. In this lecture, we will be discussing about the definition of chronic inflammation, characteristic feature of the chronic inflammation, causes of chronic inflammation, and chronic inflammatory cells as well as their mediators. So according to the Robbins basic pathology, chronic inflammation is defined as an inflammation of prolonged duration, which lasts from weeks to years, in which continuing inflammation, tissue injury, and healing, which is often by the fibrosis, proceed simultaneously. So what does this definition is trying to tell us? This definition tells us that chronic inflammation is inflammation of prolonged duration, which lasts for weeks to years and is a continuous process. Inflammation, tissue injury, and healing is occurring at the same time. How is chronic inflammation characterized? It is characterized by three important points. Infiltration with mononuclear cells. These mononuclear cells includes macrophages, lymphocytes, and plasma cells. Tissue destruction, which is induced by the products of inflammatory cells, and repair, which is characterized by angiogenesis and fibrosis. How does the chronic inflammation arise, or what are the causes of chronic inflammation? So chronic inflammation arises in these following settings. Whenever there is a persistent infection, this infection elicits T lymphocyte mediated delayed hypersensitivity reaction. For example, in Mycobacterium tuberculosis, there is a prolonged persistent infection which leads to the chronic inflammation. Another example for persistent infection is Treponema pallidum, which causes syphilis. Hypersensitivity, hypersensitivity disease such as autoimmune disease, allergic disease, prolonged exposure to the potentially toxic agents, either exogenous or endogenous, such as silicosis. Silicosis is a exogenous substance that may induce the chronic inflammation. So silicosis is due to the prolonged inhalation of the silica dust, and endogenous substance such as atherosclerosis due to the excessive production of and tissue deposition of the endogenous cholesterol and other lipids. What are the chronic inflammatory cells and mediators? So there are five types of different chronic inflammatory cells: macrophages, lymphocytes, eosinophils, mast cells, and the plasma cells. So what are the role of macrophages in the chronic inflammation? Macrophages are the dominant cells of chronic inflammation. There are two sources of macrophages, one from the circulating blood monocytes and one is from the tissue macrophages. Macrophages are the component of reticuloendothelial system. What is the role of macrophages? So main role of macrophages is phagocytosis and pinocytosis. Phagocytosis is also known as cell eating and pinocytosis is also known as cell drinking. These macrophages releases various type of the biological active substances such as proteases. In proteases, enzymes collagenase and elastase are the enzymes secreted in majority. So these enzymes degrade the collagen fibers and elastic fibers. Another important chemical that is produced by the macrophage is plasminogen activator, growth factors, cytokines, and free oxygen radicals. So these free oxygen radicals are cytotoxic in nature. So what are the source of tissue macrophages? In liver, the macrophages are known as Kupffer cells. In spleen and lymphocytes, macrophage cells are known as sinus histiocytes. In central nervous system, microglial cells are the macrophages. In lungs, alveolar macrophages are type 2 pneumocyte cells. In placenta, Hofbauer cells are the macrophage cells of placenta. Osteoclast cells are the macrophage cells of bone. Germinal center of lymph nodes, macrophage cells are tingible body cells. In skin, macrophage cells are Langerhans cells or dendritic histiocytes. In glomerulus, mesangial cells are the macrophages. So this slide is very important for the competitive exam. So macrophage cells are frequently asked in the MCQs. So discussing about the lymphocyte. So lymphocyte is mobilized in antibody mediated and cell mediated immune reaction. So in chronic inflammation, usually immune mediated chronic inflammation, lymphocytes are mobilized. T and B lymphocytes migrate in the inflammatory sites and releases the chemokines. Lymphocytes and macrophages interact in a bidirectional manner. So what does this line is trying to tell us? That macrophages and lymphocytes both play important role in the chronic inflammation. Whenever the activation of macrophages occurs, macrophages releases different type of the cytokines that stimulate the activity of the lymphocytes, and these lymphocytes release a further chemokines and cytokines that activates or stimulates the function of the macrophages. So both works together to promote the chronic inflammation. Lymphocytes plays important role in chronic inflammation. So B lymphocytes plays the role in the antibody production, and T lymphocyte cells plays role in delayed hypersensitivity reaction and cytotoxicity. Another mediator of chronic inflammation are plasma cells, which are derived from the activated B lymphocytes. These activated B lymphocytes produce antibody directed either against the persistent antigen in the inflammatory site or against altered tissue component. So antibodies are produced against the persistent antigen as well as the altered tissue component. Plasma cells are the larger than lymphocytes, and these cells have abundant cytoplasm, and it is also characterized by the eccentric nucleus with the cartwheel pattern of chromatid. Another chronic inflammatory cell is eosinophil. Eosinophils is present at the inflammatory site around the parasitic infection. So eosinophils activity is raised during the parasitic infection or as the part of immune reaction mediated by the IgE. So whenever there is the IgE mediated hypersensitivity or the immune reaction, eosinophil counts is raised. It is also associated with allergy. So in the allergic conditions, eosinophils plays the important role. Recruitment of the eosinophils involves extravasation from the blood and their margination into the tissue by chemokines. So recruitment and extravasation of eosinophil is in the response of chemokines. A specific chemokines is present for the recruitment of the eosinophils, that is eotaxin. So eotaxin is the chemokine that helps in the extravasation of the eosinophils. Last but not the least cell is mast cell, which is also known as the sentinel cell or was cells, which is widely distributed in the connective tissue throughout the body. Mast cells are present throughout the body and is scattered in the connective tissue. Mast cells participate in both acute and chronic inflammatory response. These mast cells elaborate cytokines, that is, it produces cytokines such as tissue necrotic factor and chemokines. Mast cells are active in the atopic individuals. So individuals which are more prone to the allergic reaction, there is high number of the mast cells in the atopic individuals. Mast cells are armed with the IgE antibody. So IgE mediated immune reactions is also stimulated by the mast cells. As the environmental antigen enters, it releases the histamines and arachidonic acid metabolites, which leads to the anaphylactic shock. So anaphylactic shock is also due to the mast cells. So this much is for chronic inflammatory lecture. Thank you.